The overall goal of this procedure is to isolate specific cell populations from the developing cerebellum. This is accomplished by first harvesting cerebella from neonatal mice carrying fluorescent proteins. The second step of the procedure is to obtain live tissue sections using a vibratome. The third step is to microdissect the external germinal layer of the cerebellum under a fluorescent dissecting microscope. The final steps are to dissociate the tissue into a single cell suspension and collect cells by fluorescent-activated cell sorting. Ultimately, results can show that specific cell populations can be isolated from the cerebellum.
The main advantage of this technique over existing methods, such as laser capture microdissection, is that cells are living and can be cultured for further experimentation.
For this procedure, sterilize the surgical tools using an autoclave. Get fresh or reheated 3% low-melting agarose ready to use in a 37 degrees Celsius water bath. Make up the required solutions for the papan-based cell dissociation. Prepare fresh NBB27 culture media. Then filter sterilize and store in an incubator. Prepare the fax buffer.